Hello, everyone, and welcome to the 7th event in our MetroFlow Corporate Member Launch Seminar Series. I'm Christina DeCoste, Director of the Flow Cytometry Resource Facility at Princeton University and President of MetroFlow, the New York, New Jersey Flow Cytometry Users Group. Since we weren't able to meet in person in 2020, the 15 members of our MetroFlow Steering Committee created this launch seminar series as a vehicle for our corporate members to reach our MetroFlow audience via the virtual platform. This session is the 7th in the series. You can find our previous online events archived on our YouTube page that you can link to through our website, metroflow.org. As a disclaimer, MetroFlow remains neutral and in no way endorses any one vendor or their offerings over another. All corporate members have had the opportunity to submit a proposal to present in this launch seminar series, and the purpose of these events is to showcase the innovations in the field that we can all take advantage of to better support our scientific and research endeavors. We hope that all of you will be able to take what you learned from this seminar series back to your labs and that the information will be helpful to all of those who are joining us today. Please use the Q&A button at the bottom of your screen to submit questions. The questions will be read to the speakers at the end of the presentation by our MetroFlow VP, Kathy Daniels. Please use the chat function to report any technical difficulties. Before we get started, we'd like to thank today's speaker, as well as you, our MetroFlow members, and the extended audience for your time and participation. And with that, I'd like to introduce our speaker, Dr. Brian Baris, sorry, I did get that wrong. Dr. Brian Baris, Technical Sales Manager at AKADEUM Life Sciences, who will be telling us about their buoyancy activated cell sorting microbubbles. Thanks, Brian. You can go ahead and share your screen.
Thank you, Christina. I appreciate that. Let me get this launched. Okay, can you guys see the screen okay? And can you hear me okay?
Yes, on both.
Excellent. Thank you very much. All right. Well, thank you for the kind introduction and a big thank you to everyone for joining this afternoon. It's a true pleasure to be able to showcase our unique technology with the scientists that are on the front lines of discovery. So as some of you are working from home, as I am, I hope you understand if we run into any technical issues or you hear any background noise, such as from my cats or my dogs. I apologize for that if you hear anything. We do appreciate your patience and involvement. Please screen capture or write down our contact information. Should you have any questions that don't get answered this afternoon, you can easily reach out to me and I'll promptly respond. My contact information will be displayed again at the end of the talk. I will shut off my video now and we'll get started into the presentation. For today's presentation, I will open things up by talking to you about the importance of sample preparation in scientific workflows, and we'll introduce you to AKADEUM Life Sciences buoyancy activated cell sorting microbubble technology. I will describe the BAX technology and demonstrate how it can be used to make your isolations quick and painless. Then I'll dive into a technical discussion involving our newest creation, the dead cell depletion microbubbles, our innovative approach to dead cell removal using microbubbles, and walk you through the associated performance data. I will then describe our second product feature, the mouse RBC depletion microbubbles, and walk you through some associated data with that as well. I will then wrap up with a quick summary of today's takeaways, followed by a discussion session. So I'm hoping by that point, you will be as excited by our innovative microbubble technology as we are. So who are we? AKADEUM Life Sciences is a private early stage life science company based in Ann Arbor, Michigan. Founded in 2014, AKADEUM was established with the goal of advancing human health. So AKADEUM creates advanced isolation products and fundamentally changes the way that isolating chemical and biological targets is approached. Our goal is to enable entirely new assays and workflows by delivering a microbubble platform technology that can isolate any sample, any volume, anywhere. So sample preparation is a critical first step to any scientific workflow. I'm confident that the majority of those in attendance today will attest to the fact that not a large chunk goes into running the experiments, but it's actually spent preparing their precious samples for the day's experiments. This process is often arduous and time consuming. It generally starts with obtaining a sample, let's say a blood sample for the purposes of this presentation, although most often a researcher is typically interested in a very particular part of that sample, whether that be DNA or RNA for genomic or transcriptomic studies, proteins for those interested in proteomics or understanding cell signaling pathways, or entire cells for single cell sequencing or flow cytometry. Without some form of sample cleanup or purification, it's usually pointless to analyze those samples, simply because there's so much background noise in the sample that it becomes impossible to obtain accurate and meaningful results. And in some cases, it's perhaps even dangerous and costly if you're making decisions based off of those findings without even recognizing the issues. So welcome to the next generation of cell isolation technology with our revolutionary buoyant approach using BAC's microbubbles. Our gentle yet precise technology allows researchers a new choice when considering cell enrichment or depletion methodologies. Our technology will maintain target cell health and physiology while delivering performance unmatched by existing technologies. Our buoyancy activated cell sorting, or BACS, is AKADEUM'S patented approach to separation and offers a novel way to isolate targets of interest, and that can include cells, nucleic acids, proteins, chemicals, and actually much more. We functionalize tiny floating particles that we call microbubbles and develop the protocols and devices to put our microbubbles to work. Our technology combines the specificity of affinity molecules like antibodies with the power of gravity in a workflow that's fast, easy, and effective, while being exceptionally gentle on delicate targets like rare immune cells of low abundance. But what exactly are microbubbles? So the core of each microbubble is a hollow microsphere that allows researchers to leverage the power of gravity for quick and gentle removal of captured targets from a complex liquid mixture. AKADEUM'S microbubbles are on average 16 micrometers in diameter with a polydispersed mixture and are strong and stable. The microbubbles have a density of about 0.6 grams per centimeter cubed, which allows them to float in water, PBS, culture media, biological fluids, or any other aqueous lab solution. Their density allows them the power to actually lift multiple cells per microbubble, but does so gently so as to minimize shear forces on delicate annulates. AKADEUM'S microbubbles can be functionalized in a variety of ways to allow for capture of diverse annulates. For example, the microbubbles can be strep-avidin coated to enable isolation of biotinylated targets, antibody coated for immunopurification purposes, oligo coated for capturing complementary nucleic acids, or developed using customized methodologies to suit specific research and biotech needs. The flexibility of the microbubble platform gives researchers the ability to quickly and easily isolate and concentrate cells, DNA, RNA, proteins, chemicals, and much more using a simple workflow that is inherently customizable. So with gravity as the driving force powering the microbubbles, they deliver consistent results every time, enabling reliable separations that are not dependent upon sample volume or container shape or size. Microbubbles can be used on any sample of any volume in any container without any problems. It's truly remarkable. So our microbubble technology is so elegant in simplicity, yet powerful in application. So we are the provider of the next generation of cell isolation technology. So now let's dive a little bit deeper to ensure everyone is familiar with how our technology actually works. So our buoyant approach to separation eliminates many of the existing technical hurdles that are limiting improvements or effectiveness of processes today. Our microbubbles do not have volume or equipment restrictions like you may be accustomed to having. AKADEUM'S microbubbles are gently mixed into the sample where they engage the targets before isolating those targets through flotation-based separation. It's literally that simple. So as you can see in this time-lapse demonstration, a gentle mix is all that's needed. The microbubbles, using their inherent buoyancy, will simply float to the top of the container, bringing with them their bound targets. We've taken technology that traditionally requires additional equipment and consumables and put it into a single container for self-separation. No magnets, no columns, no limitations. Okay, now that we got that out of the way and we've covered the basics, I'd like to transition over to our first topic that we are here for, dead cell removal or dead cell depletion. I may use depletion and removal interchangeably. I apologize for that, but it means the same thing. The need for an efficient and gentle elimination of dead or dying cells is widely understood. So let's take a moment to remind ourselves of the impact dead cells have on 4 different applications. We'll start with single cell sequencing. So this application has broad applications across biomedical research. It has quickly become one of the most powerful techniques for investigating complex biological systems at a single cell resolution. When using single cell RNA-seq, it is critical that only single viable cells are included in the analysis, as the presence of compromised cells can lead to a misinterpretation of the collected data. So sample preparation to remove dead cells before single cell analysis not only improves library cleanliness and complexity, but critically improves the rate of cell recovery. This leads to higher quality genomic data as a result. Current protocols for isolation and preparation of cells for single-cell RNA-seq are often produce stressed, damaged, or dead cells. It's common for these cells to have issues with the integrity of their cellular membrane, causing them to leak DNA or RNA into the surrounding environment. These nucleic acids released from the cells damage tissue dissociation or process steps contribute to higher background noise and contaminate the actual data. For these reasons, protocols and best practices for single-cell RNA-seq equipment providers like 10x Genomics recommend removal of dev cells prior to sequencing in order to obtain high-quality results. Let's slide over to the next one. Now we're going to look at target cell isolation. So dead cells even play a role in targeted cell isolation. These dead cells can lead to negative downstream impacts, including poor sort efficiencies, extended sort times, and overall reduction in purity. Dead cells can aggregate and adhere to healthy, viable cells, thereby reducing the overall count of useful cells obtained from a sample. And furthermore, these aggregate cell clumps can clog cell sorters, creating the need for additional steps to remove or break up the clumps prior to sorting. These steps may include DNA digestion, trypsinization, filtering, or extra washing, any of which may have a negative impact on the final cell counts and viability. So emerging immunotherapies, including those using the chimeric antigen receptor T cells or CAR T cells, rely on efficient purification and culturing of those T cells. But this can be incredibly challenging when it comes to maintaining T cell viability and effectively avoiding cell death. This type of process requires workflows and technology that allow for sample preparation to enrich for target cells while minimizing cell death and the negative impacts thereof. T cells and CAR T cells can become overstimulated during processing, which can lead to activation-induced cell death. Furthermore, differences in enrichment methods or approaches to cell culture can also lead to an increase in cell death and a decrease in the final viability of the cultured cells. Optimized dead cell removal or depletion could help to overcome the current limitations and technical barriers that are preventing cutting-edge therapies like CAR T from being realized, as DCR could aid in producing therapy-grade T cells for use in research, contributing to scientific advancements in this area. Finally, there's also evidence that dying cells can impact the activity of neighboring healthy cells. I think we all know this. So dying cells have been shown to produce factors like TGF-beta, IL-10, lactoferrin. Not only do effects facilitated through the release of these soluble factors influence nearby healthy cells, but dead cells and their remnants, including membrane fragments, DNA and RNA organelles, cytoplasmic contents, and other intracellular components can interact directly with viable cells. Together, these soluble factors and interactions with cellular remnants can directly impact the metabolism, proliferation, activation, differentiation, or even viability of the healthy cells encountering them. So now that we've got that out of the way and we fully understand the impact of dead cells, let's get more into the actual product itself. And for this, I'm going to walk you through a tale of dead cell depletion and how we actually got to the position we find ourselves in this afternoon. It's a tale from partnership to product. So the dead cell depletion application showcases a perfect example of how partnering with AKADEUM can turn a great idea into an innovative tool. Because sample preparation and analysis is frequently complicated by the presence of dead cells, leading to negative downstream impacts like poor sort efficiencies, extended sort times, and overall reduction in purity, an idea was posited by one of our partners where they suggested a simple way of removing dead or dying cells could be to use our strep-avidin microbubble kit. Our client was aware that there are multiple ways of removing dead cells, but none could achieve high recovery and high viability consistently, so they figured there must be a better way. Enter AKADEUM microbubbles. Depletion of dead or dying cells could be achieved through the selective capture of cells exposed phosphatidylserine using a Nexus 5 conjugated Bax microbubbles. We supplied our partner with the SA kit. We worked with them on many different protocol iterations. And from the successful proof of concept, AKADEUM'S R&D team went to work on developing and optimizing the DCD application. But going back to that bright idea posited by our client for potentially using microbubbles to target dead cells, can it actually work? With the knowledge that our technology can enable samples previously discarded, we figured an application like dead cell depletion is right up our alley. AKADEUM'S microbubbles are equipment-free and can enable both low volume and large volume separations. Our sensitive and specific microbubbles enable effective capture of targets of low abundance while eliminating issues with non-specific binding. Critically, The microbubble workflow is exceptionally gentle, which is especially valuable when enriching for delicate targets. With microbubbles, throughput is never limited, which enables sample preparation that can be done at scale. So I think everyone in the audience would agree that if all the existing benefits of microbubbles that you can see here on this slide are translated into a dead cell depletion kit, we would be on to something that will dramatically enhance sample preparation, resulting in better, more relevant data. So using our microbubbles for depletion is simple. As seen in panel 1, the pre-conjugated microbubbles are mixed into the sample where they encounter and bind to their target cells. Once the sample has been exposed to the microbubbles will carry the bound cells to the surface, and an optional centrifugation step can be used to pellet the untouched cells of interest. Finally, vacuum aspiration removes the microbubble bound cells and supernatant, leaving a highly enriched population for downstream applications. So the chart on the right on this slide describes the workflow for how we got where we are today and is the agenda for the performance data for our dead cell application. From validating and proving the concept to optimizing and iterating. And finally, we will compare our BAX microbubble approach to both column-free and column-based magnetic-based DCR applications. Now, let's look at the mechanism of action for the DCD application. Apoptosis, the natural process of cellular death, is defined by a series of biochemical events that lead to a characteristic change in the cell. During apoptosis, phosphatidylserine, a phospholipid normally found on the inner surface of the plasma membrane, is redistributed to the extracellular surface. This relocation of PS to the outside of the cell membrane functions as a cellular marker of apoptosis, making it an effective target for identifying dead cells within a biological sample. The exposed PS is able to engage with a number of molecules, including annexin 5. It is this PS annexin 5 interaction that can create an effective mechanism by which dead cells can be removed. Okay, I've teased you guys long enough. Let's dive into the performance data and see how it actually works and performs. So I'm sure that this audience is all too familiar with the tedious and time-consuming nature of column-based isolations. As shown here, the workflow for column-based magnetic removal of dead cells involves the addition of microbeads, a 15-minute incubation, wetting of the column, passage of labeled sample through the column, followed by not one, but 4 more washes through the column before possibly being ready to centrifuge and resuspend your valuable cells. Everyone would agree that this is a lengthy and involved process. While the column-free workflow is faster and easier than that of the column-based magnetic approach, it still involves pipetting of multiple reagents, requires multiple incubation steps, and finishes with the dreaded blind pour. We at AKADEUM believe that there is a better way. So our BAX microbubble approach is quick and easy. It involves one pipetting step in which the DCD microbubbles are mixed into the compromised sample, one incubation step where the microbubbles seek out and bind to the dead and dying cells. The separation of the microbubbles and capture cells actually happens at the same time that you're pelleting the untouched viable cells. And these microbubbles, captured cells, and supernatant are all removed in one vacuum aspiration step. I know it's hard to believe, but it really is just that easy. Our simplified protocol is easy to follow, offers unmatched processing times, and I'll show you through the rest of this webinar they perform outstandingly well. So over the course of the slides that I'll present today, I'll walk you through the evolution of AKADEUM'S dead cell depletion microbubble platform. As you can see from the indicator bar at the top of the slide, we will be starting with data from our initial proof of concept studies. So for these initial proof of concept studies, Jurkat cells were cultured overnight at 4 degrees C to induce apoptosis. The following day, AKADEUM DCD microbubbles were used to deplete dead and dying cells. Viability was assessed using annexin 5 and 7 AAD staining. Looking at panel A, you can see the flow plots for before and after the depletion. In these plots, the 7 AAD is indicated on the y-axis, while annexin 5 is indicated on the x-axis. As a reminder, annexin 5 bonds to the phosphatidylserine that is accessible due to the loss of membrane integrity or having been flipped to the outer membrane surface as apoptosis is induced. So the healthy viable cells in these plots are located in the lower left quadrant. The density plots demonstrate an enrichment of viable cells following microbubble-based dead cell removal. While these plots only show results for an individual sample, the table in panel B summarizes the results for 3 independent experiments. Another key point indicated by the table in panel B is that the depletion of dead cells occurred with minimal non-specific loss of viable cells. Since one of the biggest challenges facing single-cell RNA sequencing experiments is interference from contaminating ambient RNA, and dying cells can leak RNA into the environment, the effect of microbubble-based DCD on ambient RNA concentrations was determined. Decreasing ambient RNA during dead cell removal processing could greatly improve resolution of single-cell sequencing experiments. To measure the impact of microbubble-based processing on ambient RNA, the supernatant was collected from several jerkat samples, both before and after the microbubble-based dead cell removal workflow being performed. RNA was then extracted from these supernatant samples and quantified. Panel C indicates that along with the efficient removal of dead cells, the DCD microbubbles greatly reduced the levels of ambient RNA contaminating the sample. AKADEUM'S DCD microbubbles were used to deplete dead and dying cells in 3 unique samples that included jerkat cells, human PBMCs, and mouse splenocytes. The percentage of dead cells removed was determined based on the number of dead cells in the cleared sample relative to the number in the starting sample, and 3 independent replicates were performed for each sample type. AKADEUM'S dead cell removal microbubbles cleared over 80% of the dead cells across all 3 sample types. These early results gave us confidence to pursue larger studies and work on iterating and optimizing our protocol. So given the fantastic early results that we obtained, we wanted to perform more robust testing and get an idea of how our protocol actually compares to our magnetic competitors. So here the data represents processing of 10 samples with AKADEUM dead cell depletion microbubbles or a column-free magnetics competitor. AKADEUM'S DCD microbubbles were able to effectively and efficiently remove dead and dying cells in both jerkat cells and human PBMCs, yielding final viability numbers that are comparable to column-free magnetics. Now, if you remember a couple slides ago, I mentioned the ability of the microbubble protocol to remove dead cells with minimal loss of viable cells. We were very excited by that finding because one of the major complaints with current dead cell removal options is that too many cells of interest are lost through the dead cell removal process, which severely limits the researchers' ability or what they're able to do with those enriched cells. So panel F shows recovery data for viable CD45 positive cells following dead cell depletion. This data was obtained from the same sample that we just discussed in panel E. As the bar graph indicates, the AKADEUM dead cell depletion microbubbles led to the recovery of significantly more viable cells than were in the starting sample. Now, we were very excited by how well the microbubbles performed, but the icing on the cake came when we compared the time it took to process 10 samples using AKADEUM'S dead cell removal microbubbles or the column-free magnetics protocol. Not only did we achieve equivalent final viability numbers, but the microbubble platform improved the yield dramatically and did so faster than the column-free magnetics. So now, over the course of the next couple slides, I want to explore further comparisons of AKADEUM'S microbubble platform versus column-free magnetics. In these slides, we will move into mouse splenocytes as the sample type. This slide highlights the enrichment of viable cells across the samples with a moderate or low starting viability. Again, the viable cells are those shown in the lower left quadrant. The plots indicate that the dead cell depletion microbubbles and the column-free magnetics kit offer enrichment of viable cells across a range of starting viabilities. These plots also hint at a decrease in recovery of viable cells when using magnetic approaches. I'll explore that further in the next 2 slides. So now these tables summarize results from 6 independent samples and indicate that the dead cell depletion microbubbles and the column-free magnetics kit offer comparable enrichment of viable cells across a range of starting viabilities. For the moderate viability samples, the average starting viability was about 77.9% and was improved to 91.1% with AKADEUM'S microbubbles and 92.7% with column-free magnetics. For the low viability samples, the average starting viability was a meager 37.2% and was improved to 85% with AKADEUM'S microbubbles and 83.2% with column-free magnetics. Taking this a step further, we see that once again, the microbubble platform really shines when looking at the number of viable cells that are retained throughout the dead cell removal process. In this instant, AKADEUM recovered 5 times more viable cells. That is an enormous difference. The next 3 slides contain data from jerkat cell samples that were processed using the AKADEUM microbubbles or column-based magnetics protocol. In this set of experiments, the average starting viability was just shy of 40% and was improved to 74.8% with the AKADEUM platform, but only achieved 65.1% viable with the column-based magnetics protocol. So similar to what was shown for the column-free magnetics, the column-based magnetics protocol also led to a significant loss of viable cells that were in the starting sample. Here, Acadium performed over 2 times better than the column-based magnetics. Further highlighting the troubles of column-based enrichment is the time required to process those samples. So as we discussed at the onset, the column-based magnetics approach had the longest and most tedious protocol, and that is borne out in the processing times. We calculated the time required to process 6 samples with each kit and found that processing 6 samples took, on average, 25 minutes with Bax Microbubbles and 58 minutes with the column-based magnetics kit. So to summarize the findings that were presented here for DCD, I'll leave you with this table. As you can see, all three methods we compared are capable of enriching viable cells. However, Acadium's dead cell depletion microbubbles were able to accomplish that enrichment faster and recovered significantly more usable cells. So with the results that we're seeing from the dead cell depletion microbubbles to date, the natural question is, well, what's coming next? So there are 2 key things to watch out for as we continue to develop our dead cell depletion approach. The first is that we're continuing our iteration and optimization approach and are fine-tuning our protocol for an expanded range of sample types. If dead cell contamination is causing you headaches, I want to hear from you. We're looking to learn more about your samples where dead cells are creating roadblocks for you and to hear more about the hurdles you're running up against so that we can ensure that our dead cell depletion microbubbles are solving your biggest problems. The second thing to take note is that we've launched an early access program for select researchers. If you're interested in being part of this program, please do reach out to me and I'll keep in close contact as we add new researchers to this program. So speaking of the early access program, thus far I have received data from samples such as Drosophila germ cells, mouse lung and uterus, human nasal tissue, and the list goes on. I have about 6 other samples that I haven't spoken of yet. In each case, our microbubbles were able to rescue all the samples regardless of their starting viability. This kit has proven to enable samples that were otherwise discarded due to recommended guidelines regarding viability and dead cell counts for downstream applications like single cell sequencing. We were confident in the beginning with our R&D data, but after seeing the performance of the DCD microbubbles on real raw samples, I can now advise you that this is by far the best dead cell removal system ever created. The data on this slide comes from one of our early access customers at the Whitehead Institute of MIT and Harvard and the associated floor core expertly managed by Kathy Daniels. On the left, you can see that without microbubbles, the sort efficiency was 72%, with the sort time taking over 43 minutes. After applying the dead cell depletion microbubbles on the right, you can see that the sort efficiency improved to 93%, with the sort time just taking over 6 minutes. We are expecting a full product launch early next year for DCD microbubbles, but we do plan to keep the early access program alive until then. If anyone has interest in learning more, or if you'd like to be added to the list of interested researchers, please do let me know. Okay, now that we've covered DCD in good detail, let's transition over to mouse RBC depletion kit. This kit performs in a similar fashion as the DCD microbubbles, but instead of recognizing annexin 5 positive cells, they target the tier 119 antigen on mouse RBCs. Using our 3-step protocol that even with a 5-minute centrifugation step, RBC contaminated samples can be efficiently cleaned up in roughly 10 minutes. So looking at the flow plots, in the top right corner, we see RBC contaminated PBMCs before and after an Acadium depletion. While it is clearly plain to see that the RBCs have been removed just by looking at the cell pellets before and after depletion, the FACS plots that you can see do help us see just how efficient that depletion was. So in this example shown here, we see that roughly 46% of the initial sample was RBC contamination. However, following the microbubble-based depletion, that percent drops to just 5%. So let's take that a step further. So we can clearly see that the impact that a sample cleanup as powerful and convenient as the Acadium red blood cell depletion can have. This example depicted on the table that you can see in the bottom right corner, CD19 B cells were sorted from PBMC preps before and after RBC depletion. The time to isolate 250,000 B cells was determined and the results are pretty clear. Using RBC depletion kit, cut over a third of the time off of these sorts. This is already a huge benefit, but now imagine what that could mean for isolating rare cell types. It could truly be the difference that enables projects that would otherwise not be feasible. While the labor and cost savings are probably enough for most, another key factor to consider is the stress inflicted on cells that must be out and waiting for the sort to be completed. Reducing that time allows for the collection of healthier cells that greater reflect the experimental conditions. Now let's examine another use of the mouse RBC depletion kit as a tool for enabling the isolation of these rare cell types. So an effective sample cleanup is especially important when dealing with low abundant cell types. Invariant natural killer T cells, or iNKT cells, represent one such cell type. iNKT cells are a subset of unconventional T cells that recognize lipid-based antigens and have been shown to have a potent anti-tumor response when stimulated appropriately. With iNKT cells representing less than 1% of splenocytes or cells in peripheral blood, it can be quite difficult and time-consuming to isolate enough high-quality cells to perform the studies necessary for advancing our understanding of their potential. In collaboration with our customer at the Albert Einstein School of Medicine, we developed a method for depleting both erythrocytes and B220 positive B cells from mouse spleens prepped using AKADEUM'S BAX microbubbles as a mean for enriching low abundance T and iNKT cell populations. So the mouse RBC and B cell depletion microbubbles removed over 95% of the RBCs and over 93% of the B cells. So let's go a little bit into the data on this slide. So spleens from humanized mice were removed, dissociated, and erythrocytes were removed using either RBC lysis buffer or AKADEUM'S mouse RBC depletion kit. So looking at the top, the graph on A, representative flow plots depicting the percentage of tier 119 positive erythrocytes following an RBC lysis buffer treatment on the left, or after depletion of tier 119 positive cells using AKADEUM'S mouse RBC depletion kit on the right. You can see that a bar graph depicting the percentage of tier 119 positive cells across individual samples with an N of 6. You can see that this data demonstrates an average depletion of greater than 95% of tier 119 positive erythrocytes, yielding a final purity greater than 97%. Now looking at the bottom data on graph A, we see a depiction of the percentage of remaining RBCs and debris in samples that were treated with lysis buffer or had RBCs removed with AKADEUM microbubbles. While the removal of RBCs was shown to be similar, this graph demonstrates that using AKADEUM'S microbubbles to remove RBCs left samples with less residual RBCs and debris. We had an N of 12 in this experimental subset. Moving over to B on the bottom there, we see a bar graph depicting the percentage of viable cells following the lysis buffer treatment or microbubble-based RBC depletion. You can see that this graph demonstrates a trend toward better viability when using our gentle approach instead of the harsh lysis buffers. So sequential application of these simple 10-minute sample cleanup protocols saved time and resources, improved sample quality, and led to more meaningful results. So this work displays the unparalleled adaptability of the AKADEUM platform and clearly demonstrates the value of AKADEUM'S easy, gentle, and effective nature. And finally, I would like to showcase one more example of how our RBC depletion kits can be used in conjunction with other microbubbles for a multi-targeted depletion approach. In this example, I will show how circulating tumor cells, or CTCs, were enriched from a simulated liquid biopsy sample. The CTCs were enriched from a starting sample containing 49.5% CD45 positive cells, 49.5% CD235A positive RBCs, and 1% cancer cells through a targeted depletion of CD45 positive and CD235A cell types. As you can see from the table, greater than 99% depletion was achieved for both cell types, and CTCs were enriched about 75 times. These incredible results showcase how powerful our microbubbles are, but I also hope I conveyed to you that our microbubbles are by far the most gentle and effective cell separation platform that exists. So dead cell depletion and mouse RBC depletion are only two of the most recent applications where AKADEUM'S microbubble approach to separation is disrupting the status quo. AKADEUM'S microbubble technology is the next generation in separation techniques, and its inherent flexibility allows for targeted isolation of any number of targets, everything from cells to nucleic acids to proteins or chemicals. Our elegant and easy to use microbubble approach enables faster, more accurate workflows with exceptionally gentle processing that increases the throughput and allows for greater recovery of rare cells. Our microbubble platform is also uniquely scalable in both the number of concurrent samples being processed as well as in fluidic volume. And key to all this is our team that is committed to your success. We thrive on problem solving and are dedicated to helping the researchers we work with see exceptional results. You can talk to me or any one of our scientific support team directly about your application. We are here to help you overcome the long-standing headaches in sample preparation. So to conclude, I'd like to invite everyone to consider a simple thought experiment. Do you have an existing hurdle in your research where our microbubbles could be the solution? Or perhaps you have an idea for a product that can solve a problem. Regardless of what it is, if you have an idea, I want to hear about it. Please reach out to me and I'll help you explore the feasibility. Thank you all for your time and attention. I'd like now to open up the discussion by taking some questions from the audience. If we run out of time before we get to all questions, please feel free to email me your questions and I will promptly respond with an answer. I would like to remind everyone to please write down or screenshot my contact information from the slide for easy access later if you happen to have any questions or would like to get a microbubble kit. Thank again to their attention and for the organizers of MetroFlow. I really appreciate this opportunity. Now I'm happy to address any questions.
Okay, wonderful. Thank you so much, Brian, for a really great talk today. We're really happy that you're able to talk to us about the BACS technology. So we do have a couple of questions and I'm going to get started with the first one. So the first question that probably comes up fairly often is, and I think you showed some of this data, but we still might have some people curious, how safe are these microbubbles to use in combination with cell sorting technology? So Flow Core Manager, we don't want to put anything through our instruments that are going to do any damage. So if you can give us some input on that, that would be great.
Yeah, so this is probably the #1 most requested question that we get. Is it safe for microbubbles to be put into instruments? And the answer is unequivocally yes. So we've even used microbubbles in microfluidic devices and they performed phenomenally well. I believe that, because of the size of the microbubbles being a mixture with the average size around 15 micrometers, they're definitely small enough to easily pass through. If any clumping occurs, it's typically related to the cellular sample itself and not the microbubbles, and simple cleaning techniques and procedures could be done to keep the instruments safe. But yes, they are completely capable of being put through a number of instruments and devices without any cost or concern.
Okay, wonderful. We have another question that came through to ask how involved the process is to optimize the microbubble to cell number ratio for each project or application. So say someone's working on optimizing an experiment, how much work goes into getting there as far as the prep goes?
So surprisingly, it's not that bad. So that's a great question. So the microbubble to cell ratio can be altered in some ways that could actually get you better performance data. So from the application of dead cell removal or dead cell depletion, if you guys have very low starting viability, say in the 20s or 30%, we can try and increase microbubble to cell ratio. We typically start at a 2-to-1, so 2 microbubbles for every cell. We can increase that to 3-to-1 or 4-to-1, in which case you will get even more increased depletion. And this sort of this step or optimization can be done in any sort of isolation or depletion with our microbubbles. It doesn't take that long. Honestly, with our team in place, we've worked with the microbubbles on a number of different samples, most in fact. And so we can give you advice and help you decide which is the best to test out first, because we have a lot of that data already. So reach out to me, I'll be able to help you with that.
Perfect. And then we have another question that had come in asking if this technology can be used to deplete cell populations such as like CD8 positive T cells.
Great question. Yes. So I didn't talk about any of our commercialized kits in this talk. I just focused on those 2 applications, but we do have commercialized kits and we do have some that are immune based. So we do have T and B cell kits. We just finished our Series B round of financing about a couple of weeks ago. And so we are ramping up significantly on the number of kits that will be out. We do have a CD8 kit that will be coming out, I'm hoping within the next few months or so, in which case it may be put into an early access program to kind of garner feedback from the group, or it could just be a full product launch and ready to go. In any case, I invite you to send me an email so I can have your information. I'll put you on the list for human CD8 and I'll let you know when it's available.
Perfect. And then along that line of thinking, something that comes up in research flow cytometry cores often is some bizarre cell types maybe, or certain targets that aren't so common. So will there be a possibility of custom depletion kits potentially in the future? So if there's a certain target that really people are interested in, is that something that you're willing to work with customers on?
That is an excellent question. So yes, we are 100%. And I'll give you a little bit of a story. So I did work with Svetlana. She is the flow core director at Rockefeller. We had this idea of depleting myelin for neuronal samples because myelin is such a heavily, it's everywhere in neuronal samples, as you can imagine. It's a very abundant protein. It clogs or confounds the data. So we had the idea of using the streptavidin microbubbles connected with an anti-myelin antibody that we'll put onto the microbubbles. And when we tested it out, it did work, but there were a number of factors that we're still trying to work out it didn't work out as best as we had hoped. However, I just want to give this example as our efforts to work with any number of customers. We're willing to work with you on any sort of depletion or a specific isolation. We do have a streptavidin microbubble kit that could be used with any number of biotinylated antibodies. So if you have a biotinylated antibody against a cell type or protein or DNA or RNA that you want depleted, removed, or enriched, reach out to me. I'll be happy to set you up with that streptavidin kit, and I'll work with you on optimizing it for your specific depletion or enrichment.
Perfect. And then I have two more questions. So one question is, say there's someone coming to work with the microbubbles and downstream for flow cytometry, and they want to make sure that those microbubbles aren't included in their analysis. You said they're about 15 microns. Some cells can be 15 microns. We expect the scatter to be different, but is there any autofluorescence of these beads that would enable us to pull them out and make sure they're not included in the analysis with flow?
Great question. So yes, we have noted that our microbubbles do provide some sort of autofluorescence, but typically you don't have microbubbles in your sample. This has only been in select circumstances when aspirating the microbubbles is a little bit of a skill that requires some practice. It's not as simple as just removing or aspirating the liquid solution. It's a combination of the microbubbles with the subnatant. And so when you remove them, some people go a little bit quick, and so they may push down some microbubbles down into their purified sample, in which case that would become contaminated with microbubbles or what was supposed to be depleted from the sample. So as long as the aspiration of the microbubbles goes swimmingly, typically you don't have any residual microbubbles left over. Now, this is a problem that we are addressing. We are developing a new, I guess you could call it a vial that will physically separate the microbubbles from the purified cells so that there's absolutely zero microbubble contamination in downstream applications. But typically it's not a common problem, and it's usually related to aspirating the microbubbles. And that's something that we can work with people on, and I can help them get better at.
Okay, perfect. And then we have a question that came through from Yolanda, and it's actually a really great question. And it's asking, how about positive selection using this buoyant activated cell separation? So when both fractions have to be used downstream, can those microbubbles be removed afterwards?
Great question. So the one caveat to our technology currently is that we do not have positive selection. Our microbubbles, the streptavidin biotin linkage is, as you guys know, probably one of the most strongest in nature. So it's quite difficult to remove those two. So we are coming out with new kits that will be taking advantage of a new way to positively select and remove the microbubbles from those cells. It's just a little tease for what's coming, I believe, early for next year. So if anybody's interested in microbubbles and using them for positive selection, do reach out to me. We are able to work with some situations currently. We don't have those tubes that will provide the physical separation, but it's completely possible that we could work with you on this. So yeah, again, please feel free to reach out to me, and I'll work with you on that specific case.
Okay, perfect. Well, Brian, I think I've exhausted the questions. We definitely had a couple that came through, and thank you so much for, actually, I'm sorry, there's one more question that came through. So, we talked about the autofluorescence, right? But have you considered engineering microbubbles to have some fluorescence that they could be separated via flow cytometry, right? So that there is some fluorescent readout from it above autofluorescence that we could use.
Yes, that is an excellent question. It's one that I've had a few people reach out to me recently for, and I'm working with them on that. So yes, we can work with you on that. We are considering ways to label microbubbles so that they could be used in specific applications with flow cytometry, so you can get a quantified number of different subsets of cells. So all of that stuff is in play, and I'm very excited to hopefully maybe early next year present again, and I'll have some of that new exciting technologies for you at that time. But currently we don't have anything yet. But if you do want to send me an email with that specific question in mind, I can send you some data for a recent experiment that we did where we look at utilizing exactly autofluorescence or defining properties of the microbubbles so that they go into a flow sorter and be used for any sort of complex analysis.
Okay, wonderful. All right. So I think now we're all good on questions. Sorry about that. And thank you so much, Brian.
My pleasure.
Yes, thank you, Kathy, for handling the Q&A and everyone who attended today. So you'll be able to find this recording if you missed anything from the beginning. It will be posted within a week or so to our YouTube page that you can get to via our webpage, metroflow.org.
Great. Thank you, everyone. Thank you to the audience. Thank you, Christina, and thank you, Kathy. I really appreciate it.
Thank you.
Thanks so much.
Have a great one.
Bye.
You too.
Bye-bye.